In this chapter, we're going to take a look at genetics and cellular function. Many of the chapters in this book discuss hereditary traits, such as blood types and hair color, and genetic disorders, such as color blindness, cystic fibrosis, and diabetes mellitus. To understand such conditions, it's necessary to have some familiarity with DNA and genes. Heredity has been a matter of human interest dating for thousands of years, but the scientific understanding of how traits pass from parent to offspring began with who we consider to be the father of genetics. That would be an Austrian monk named Gregor Mendel. He's famous for his garden pea experiments. By the late 19th century, biologists had observed chromosomes under a microscope. In the early 20th century, scientists rediscovered Mendel's work and realized the correlation between chromosome behavior and his laws of heredity. From that simple but insightful beginning, genetics has grown into a highly diverse science with many subdisciplines. It's arguably the most dynamic of all natural sciences in these early years of the 21st century. Mendelian genetics deals with parent to offspring and even larger family relationships to discern and predict patterns of inheritance in the family line. Cytogenetics uses the techniques of cytology and microscopy to study chromosomes and their relationship to hereditary traits. Molecular genetics uses the techniques of biochemistry to study the structure and function of DNA and RNA. Genomic medicine studies the entire DNA endowment of an individual, which is called the genome, and then how it influences health and disease, and then how that information can be used to prevent, treat, and cure diseases. In this chapter, we'll look at all four perspectives. Here's our expected learning outcomes for the first section. You can pause the video and read those over. Improving the microscope made it a lot easier to study genetics because it made the chromosomes visible. It could be seen during cell division that when the nucleus divides, chromosomes are split neatly in two and then distribute their halves to the two new daughter cells. With that information, scientists came to suspect that the nucleus was the center of heredity and cellular control. In the 1800s, a Swiss biochemist, Johann Friedrich Miescher, studied the nuclei of white blood cells that he extracted from pus in used hospital bandages. And then he also later studied the nuclei of salmon sperm. Both of those types of cells offer very large nuclei. In 1869, he discovered an acidic, phosphorus-rich substance that he named nuclein. He correctly believed this to be the cell's hereditary matter, but unfortunately he was never able to convince scientists in his own lifetime of this. Today we call this substance deoxyribonucleic acid, or DNA for short. And we now know that he was right. DNA is what holds our genes. Not long after, by 1900s, biochemists knew the basic components of DNA, that DNA contains a sugar, some phosphate groups, and some nitrogenous bases, but at that time did not have the technology to determine how those were put together. That wouldn't come until 1953, when Watson and Crick won the Nobel Prize for figuring out the structure of DNA. So deoxyribonucleic acid, or DNA for short, is a long thread-like molecule with uniform diameter, but it does vary in length. If we could pull the DNA out of each of our cells in the human body, it would be over 2 meters long. Hard to believe that, but if you coil DNA up very, very tightly and wrap it around proteins to make chromosomes, 46 of them to be exact in each human cell, then you would get 2 meters long of DNA. Hard to believe something so long can fit into something that is microscopic. So humans have 46 chromosomes in their cells, 23 of which come from your mother and 23 come from your father. The average DNA molecule that fits on each of the chromosomes is about 2 inches long. Over to the right, we see a picture of one of DNA's nucleotides. It's the adenine nucleotide, adenine being the nitrogenous base, deoxyribose being the sugar, and then we can see the phosphate group. Remember from our last chapter that DNA is a polymer, meaning that it's made out of repeating units. Those repeating units are called nucleotides. And just like the picture on the right of the adenine nucleotide, each nucleotide contains a sugar, the deoxyribose sugar, a phosphate group, and then one of 4 nitrogenous bases. Here we see adenine. Remember, nitrogenous bases have a lot of nitrogen in them, hence the nickname. They might also have guanine, cytosine, or a thymine nitrogenous base. Here's all 4 bases, plus an extra one that we find in RNA, pictured off to the right. Adenine and guanine, if you take a look at them, they kind of resemble each other in shape. Although guanine has a different functional group that sticks off this end, whereas that functional group in adenine sticks up over here. But they look very similar. They're called purines. They have double rings. Cytosine and thymine, plus uracil, which is found in RNA, they have just a single ring. They're called pyrimidines. Instead of saying adenine, guanine, cytosine, thymine, and uracil, you can just say the first letter for each one to shorten things up. A for adenine, G for guanine, C for cytosine, and T for thymine, and if we're talking RNA, U for uracil. In the 1950s, it was finally discovered that the molecular shape of DNA is a double helix. It's like a twisted ladder or a spiral staircase. Each side piece is a backbone composed of phosphate groups alternating with the sugar deoxyribose. And then the step-like connections in the middle are the pairs of nitrogenous bases. Interestingly enough, we find that adenine always pairs with thymine because they like to have 2 hydrogen bonds between them, and guanine always pairs with cytosine because with their molecular structure they like to have 3 hydrogen bonds between them. So the rules go with DNA-based pairing. A always pairs with T, C always pairs with G. This is called the law of complementary base pairing. It's kind of nice because if you know the order of the A, T, C's, and G's on one side of DNA, then you can always figure out the order on the other side of DNA. Because A always pairs with T and C always with G. Here, I remember on the backbones, you can see the phosphate group pictured as a yellow circle, and deoxyribose pictured as a green pentagon. By 1900s, again, the components of DNA were known to be the sugar, phosphate, and then the bases that make up DNA. By 1953, thanks to Rosalind Franklin's X-ray diffraction picture, the Nobel Prize for figuring out that DNA is a double helix went to Watson and Crick, and then Wilkins, who was one of Rosalind Franklin's colleagues, who gave the picture to Watson and Crick. Unfortunately, Rosalind Franklin was not awarded the prize with these three men because she had died of cancer, most likely due to all the work with the radiation with the X-rays. She had died of cancer by age 37. Unfortunately, they don't award the Nobel Prize to people who have passed away. Here's some genetic terminology that you'll want to get used to. What is a gene? A gene is a sequence of DNA that codes for usually a protein. It can also code for a section of RNA as well, but usually codes for a protein. And then those proteins cause your traits. So basically, the order of A, T, C's, and G's make up your genes. The genome is all of the genes in one person. It was once thought that because humans are so complex that we should have over 100,000 genes, but when the genome of a few humans were figured out in the early 2000s, it was found, surprisingly, that really we only have about 20,000 to 25,000 genes that code for all the proteins and sections of RNA in our bodies. Even plants have more genes than humans, surprisingly enough. Another surprising thing we figured out, that even though DNA is really long, like 3 billion base pairs long, that only 2% of our total DNA actually codes for useful genes in our body, for useful proteins and RNA. The other 98% is called non-coding DNA. The part that actually codes for something that's useful, those are called exons. The part that doesn't code for useful proteins or RNA, those are called introns. The other 98% does, though, play a role in chromosome structure, also maybe regulation and gene activity, or if it's no function at all, then it's called junk DNA. So if we were to look in the nucleus of a cell, we would actually find chromatin. Chromatin is a mix of DNA with proteins. When DNA gets more organized and we actually see it in its chromosome form, the 46 chromosomes of the human body, the DNA wraps and coils really tightly around these proteins. These proteins are called histones. Here we can see it in the picture off to the right. Once that DNA, which is mixed in with the chromatin, coils around those proteins, then we finally have chromosomes. In a human, we can find 46 of them. Other organisms have different numbers. Then we find that when a cell is ready to divide, that DNA actually doubles. So we go from a single chromosome to one that has kind of a doubled structure. And we can see that in the picture below here on the bottom of the right, that this is actually a doubled chromosome we're looking at. We've got the one side here has one chromosome, and then it's attached to what's called its sister chromatid right here. So this is a doubled chromosome. That chromosome is ready to go through cell division, and it can be pulled apart, and then the cell can divide, and then each daughter cell will then have a copy of each gene. Again, histones are the proteins that make up chromosomes that DNA coils around. The DNA molecule, when forming chromosomes, winds around the cluster of histones, a cluster of 8, and then repeats that pattern 800,000 times, and then that creates what's called a nucleosome. Got a picture here where we can actually see this a little bit better. Isn't it neat how it makes these little rosettes of chromosome loop or chromatin loops, which then makes the entire chromosome. The nucleosome consists of a core particle, which is histones with DNA around them, and then linker DNA, which is a short segment of DNA connecting the core particles. And it's 1/3 shorter than the DNA alone. So remember when we said at the beginning of this lecture that if we could string all the DNA together in one cell, it would be over 2 meters long. So by coiling up nice and tight, like you see here, into these little rosettes, we can shorten DNA up and we can make it fit into the chromosome of cells. We can also get it more organized this way as well, so that when the cell divides, exactly half is going to go to each daughter cell. Each daughter cell is going to get a copy. Finally, each chromosome is packed into its own little spheroid region of the nucleus. This is called the chromosome territory. A chromosome territory is permeated with channels, and this allows regulatory chemicals to have access to the genes on the chromosomes. This is the state of the DNA in a non-dividing cell. It's not a static or stationary structure. It changes from moment to moment, and that changes according to genetic activity of the cell as individual genes are turned on and turned off. Whole chromosomes can migrate to new territories as the cell develops, like, for instance, moving from the edge to the middle of the nucleus as its genes are activated for certain developmental tasks. That allows genes on different chromosomes to partner with each other in bringing about developmental changes in the cell. In this picture, we see a chromosome that's been doubled. Technically, if I just had half of this, I could say that's a chromosome. But in the picture here, it's also technically correct to say this is a chromosome. But I usually like to say it's a doubled chromosome so that we can keep things straight. So each side here is called a sister chromatid. They're duplicated, so they should have the exact copies of the same genes, so that this whole complex here can be pulled apart during cell division, giving each daughter cell copies of the genes. All right, this area in the middle here, that's called the centromere. That's where the sister chromatids join together. Then there's these little proteins on the edge. Those are called kinetochores. Those are going to be what the mitotic spindle, the little proteins of the cell that run cell division, they're going to grab onto those kinetochores and they're going to pull the sister chromatids apart. Here's another picture that might help you visualize those kinetochores a little bit better. It shows little spikes coming off of them, and you can see where microtubules would attach there and pull those apart. While DNA, we stretch it out to its 2-meter length, would have about 3 billion base pairs. It has a kind of a much smaller cousin named RNA. RNA stands for ribonucleic acid, very similar to DNA in that it has nucleotides, it has phosphate groups, but it has a different sugar. It has a sugar ribose. It also comes in a number of different forms. It's kind of like the workhorse. Sometimes we give DNA all the glory because it has the genes that code for the proteins that make us. But RNA is very important too, and it's like the workhorse that does a lot of the work to make sure that those genes of DNA get translated into proteins that make us. One of the forms that RNA comes in is called messenger RNA. It's about 10,000 bases long. Basically, messenger RNA's job is to make a copy of just a gene on DNA. It just copies that gene. That's called transcription. And then because it's so nice and small and short, it can fit through the little pores of the nucleus and then take that message of that gene copied from DNA. It can take it out of the nucleus to the ribosomes, who are usually waiting on the ER or floating in the cytoplasm. And then the ribosomes use that message from RNA, hence that's why it's called messenger RNA, and then it creates the proteins. Ribosomal RNA is important too. That's what makes up ribosomes. They kind of look, instead of being a long strand, they kind of look more clumpy. Like if you would take your two fists and put them together, think about those two fists locking together, and then a strand of messenger RNA coming through your fists reading that code within the messenger RNA. So that ribosomal RNA, which makes up ribosomes, its job is to facilitate the putting together of the amino acids that make up proteins. Transfer RNA is even a little bit shorter yet. It kind of has a clover leaf appearance. It's only about 70 to 90 bases long. Its job is to bind with amino acids that are in the cytoplasm of the cell and actually transfer those to the ribosome so that they can be delivered there to become part of the polypeptide chain that makes proteins. RNA comes as just a one-sided nucleotide chain. It's not a double helix like DNA is. And again, some of these RNA molecules have quite the different shape. Messenger RNA is more of a straight chain, which is just a one-sided nucleotide chain. Ribosomal RNA kind of looks like clumps, and transfer RNA is a nucleotide chain that's been folded in kind of a cloverleaf format. Again, the sugar in RNA is different. It has sugar ribose instead of deoxyribose. It has almost the same nitrogenous bases. It does have adenine, it has cytosine and guanine, but there is no thymine in RNA. Instead, thymine is replaced by the nucleotide uracil. And if I go back to this picture here, you can see how uracil actually looks a lot like thymine anyway. Uracil and thymine are both pyrimidines. Remember, they're a single ring nitrogen base. All right, before we go on, let's see if we can answer these focus questions. Number one, what are the three components of a nucleotide? Again, you can pause the video and see if you can answer these before I do. The three components of a nucleotide are a nitrogenous base, a sugar, in DNA it's deoxyribose, and then a phosphate group. So then the second part of the question, which component varies from one nucleotide to the next in DNA? It would be the nitrogenous base. Remember the choices? They're A for adenine, T for thymine, C for cytosine, and G for guanine. Number two, what governs the pattern of base pairing in DNA? Well, remember the rules. A always bonds with T because they like two bonds between each other, and those are relatively weak hydrogen bonds. And then C always pairs with G because they like to have three hydrogen bonds between them. Number three, what's the difference between DNA and chromatin? So Remember, DNA is our genetic information molecule, which is a polymer that consists of nucleotides. And chromatin is a mixture of DNA with histone proteins. And again, we can find that material in the nucleus of our cells. Number 4, summarize the differences between DNA and RNA. Well, they're both polymers built with nucleotides, but remember, DNA is double-sided. It's a double helix, like a twisted ladder, whereas RNA tends to have just the single side, whether it's mRNA and it's kind of just a straight single-sided chain, or if it's ribosomal RNA, it's kind of clumped in its form, and tRNA twisted into a clover leaf. Remember that DNA has deoxyribose as its sugar, but RNA has ribose, and that DNA has the 4 nucleotides adenine, cytosine, thymine, and guanine, whereas in RNA there is no thymine. Instead, that's replaced with uracil. Here's something else I'd like to see if we can do this. If I gave you this as one side of DNA, would you be able to tell me what the other side is? Remember to follow your base pair rules of A's with T's and C's with G's. I've done the first one for you. You can pause the video and see if you can figure this out. And then I'm going to give you the answer here. So first, A goes with T, T pairs with A, C with G, G with C. Then we've got 2 A's, so they'll each pair with T. 2 T's, each pair with an A, C with a G, 2 G's, each pairing with a C, and finally a T will pair with an A. Now if we could take that and add a few more billion base pairs and twist it into a double helix, cut it up and put it onto 46 chromosomes, we would have our genetic code. Here's the learning outcomes for our next section. You can pause the video and read through those. What is a gene? That question, as much as we talk about it, is actually a little bit hard to define exactly. And as our understanding of DNA has changed in time, so has our definition of what a gene is. The classic definition of a gene was that it was a sequence of DNA that codes for a certain protein. But if you think about this information, that the body has millions of proteins, and now we know that we only have about 25,000 genes, that we're going to have to change our definition. We also know now that a small percent of the genes actually code for just RNA molecules, and the RNA molecule is the finished product. And so what we found is that some segments of DNA belong to 2 different genes. So there are genes overlapping each other. So that means there's some long segments with short genes embedded within, and that there's multiple related proteins encoded by a single gene, and other unexpected arrangements. So that brings us to our current definition of a gene. It's an information-containing segment of DNA that codes for the production of a molecule of RNA that then goes on to play a role in synthesizing one or more proteins. The amino acid sequence of a protein is determined by the nucleotide sequence in DNA. Again, the genome is all the DNA in one 23 chromosome set, so that's 46 chromosomes in a human. There's about 3.1 nucleotide pairs in the human genome. That makes our DNA very long. But then cut it up and string it out over 46 human chromosomes that come in sets of 23 23 of them from mom, 23 of them from dad. And then each pair of chromosomes has the same genes, but they could have different alleles or different versions of those same genes. For instance, the gene for blood type. Mom could give you the gene for type A. Dad could give you the gene for type B. So again, those are the same genes, but different versions, just a slightly different sequence of DNA. That's called an allele. The Human Genome Project, which ran from about 1990 to 2003, helped us identify the nitrogenous base sequence of 99% of the human genome. They used some good representative humans and then figured out their genetic code, and now we have that information today. As humans, we are very similar with our genetics, like over 99.9% the same. Genomics is the comprehensive study of the whole genome and how its genes and non-coding DNA interact to affect the structure and function of the whole organism. So what we found, again, we suspected there were over 100,000 genes in a Homo sapien, but actually it's not that many. It's closer to 25,000 genes. Genes generate millions of different proteins, not the old one gene, one protein theory. A single gene can actually code for many different proteins. Genes average to about 3,000 bases long and can range up to 2.4 million bases long. All humans are about 99.99% genetically identical. That's because we evolved from a common ancestor not very long ago, so there hasn't been a lot of time for a lot of differences to build up. That's 0.01% variations that we can differ from one another with our more than 3 million base pairs that we have in our DNA. The various combinations of those single nucleotide polymorphisms or differences account for all of our human variation. Some chromosomes are very gene-rich and some are very gene-poor. We now have done what we call mapped chromosomes. We know the locations of more than 1,400 disease-producing mutations on those chromosomes. And that opens up a new door and a new branch of medicine so that we can treat people at the genetic level. The body can make millions of different proteins, all from just 20 amino acids. It's kind of like our alphabet. We have 26 letters in the English alphabet, and from those 26 letters we can make all the different words of our English language. Well, from about 20 different amino acids in our body, we can make all those different proteins, millions of different proteins. And it's only from just the 4 nucleotide bases that we find in DNA, the A, T, C's, and G's. The genetic code is a system that enables these 4 nucleotides to code for the amino acid sequence of all proteins. The minimum code to symbolize 20 amino acids is 3 nucleotides per amino acid. So what we're saying here is that every 3 nucleotides, what I like to call triplets, every triplet codes for an amino acid. If we're talking a useful gene. So a base triplet is a sequence of 3 DNA nucleotides that codes for 1 amino acid. That's called a codon. When we take mRNA and we copy that gene, and we take every 3 letters in mRNA, that is a codon, and there's 64 possible codons that are available to represent the 20 different amino acids. 61 of those codons actually code for amino acids, but a few of them are called stop codons. They actually don't code for an amino acid, but they signal the ribosome to let the protein go that it's done, to drop it off, and then let it travel through the ER and then to the Golgi to be finished. One of them, the AUG, codes for methionine. That's what we call our start codon. Every amino acid sequence for a protein begins with methionine. Okay, I've pasted in a few codon charts that show how mRNA codes for certain amino acids. I've got this black and white one here, and I've got a color one in the next slide. You can also do a Google search for amino acid charts and find different ones and find the one you like out there. So I'm going to go to this slide. So remember when we did this before, we filled in the other side of DNA before, but this time from this DNA code, what I want you to do is match it up with what would be the complementary RNA code. Because this is what happens in the cell's nucleus. DNA's double helix unzips for a short amount of time, and RNA actually makes a copy. It's called a transcription of one of the sides of DNA. So we'll say it's going to be this one here. And so what I want you to do is fill in what would be the mRNA code for this. You can pause the video if you want to do it before I do it. What matches with the T, we'll always remember our rules here. T likes to match with A. Before, when we were doing just DNA, we learned that A's go with T's, but remember that RNA does not have any T's, so instead the T is replaced with a U. So I can't put a T here, but I can put a U. C's always go with G's, so that's easy. Here we have two A's. Again, I can't put T's because RNA does not have any T's, so I've got to put U's there. T's go with A. RNA has A's, so that's good. C with G. A with T, but again, RNA does not have any T's, so put a U. And then two T's left, so that means A and A. All right, so there's our mRNA code. Now, every three of these is a codon. I'm going to do that by color so you can see them. Just picking some random colors here. Oops, that last one does not want to go. Okay, so every three of these RNAs here, or of this RNA strand, is a codon. So what we're going to do is look these up on the amino acid chart. You always use the mRNA to do that. So I'm going to look up AUG first. I'm going to go to my amino acid chart. The first base is A, so I'm going to look under the first base A. The second base is U. So here's my first base. Second base is U. I bring it down to this box. It's going to be one of these in this box. To get the third base, I go over here. Again, that codon was AUG. So here's my third base G. Bring it over to that box. And I see that my first amino acid is methionine. Going back to this slide. Instead of writing out the whole amino acid, I'm just going to write the first three letters, methionine. In fact, this other amino acid chart that I've pasted in here gives you those abbreviations because a few of them start with the same letter, so this helps us figure out what those abbreviations are. Remember, MET, methionine, is the start codon. All proteins begin with that start codon or that first amino acid, methionine. Now I'm going to look up CUU in the amino acid chart. I'll use this one this time. First base is C, second base is U. Bring it to this box. Bring the third base U over. Well, everybody in this box codes for the same amino acid. So that was pretty easy. It's called leucine, or L-E-U for short. Oops. Now I'm going to look up AAG. Maybe I'll try this chart again. Again, A, and then bring G over. Make sure I get it right. It's lysine. AAG, lysine. That's L-Y-S. I can do it over here too. AA, and then G is lysine. And I've got one more to look up. It's UAA. Let's do it on both of them. UAA. Oh, what do you know? It's a stop codon. Let's make sure here. UAA. Yep, stop codon. So either I could just leave that blank, or I'm going to write stop here. That tells the ribosome this polypeptide chain is done. It can go on to go to the Golgi, get folded, maybe get joined with some other polypeptide chains, and then it can become a protein. So that's how that works. We go from DNA's code, then we transcribe it into mRNA. mRNA is skinny enough to weasel its way out of the nucleus, head to the ribosome. Ribosome clamps onto it, and then assembles these amino acids in the correct order to make a protein. The application of our knowledge of the genome to the prediction and the diagnosis and the treatment of the disease is genomic medicine. This is relevant for certain disorders like cancer, Alzheimer's, schizophrenia, obesity, AIDS, and tuberculosis. It allows for early detection of diseases and a more effective clinical intervention. It expands the potential for gene substitution therapy, probably the medicine of the future. All of our body cells, except the sex cells and some of our immune cells, contain identical genes. Remember, half of those came from your mother, half from your father. So for the most part, 2 copies of each gene, though they might be different alleles. Different genes are activated, though, in different cells. For instance, it wouldn't be very useful for the genes of the cells of your eye to be making the protein insulin. They need to focus on making proteins that would be useful to the structure and function of your eyes. Just as your pancreas wouldn't want to be making proteins that are for the function of your eyes. They would want to be making proteins that a pancreas needs to make. Any given cell uses about 1/3 to 2/3 of its genes. The rest remain dormant and may be functional in other types of cells. So we actually have segments of DNA that act as switches that turn on genes or turn them off. Plus, the way that our genes have different functional groups added can also determine whether a gene will be functional or not. If we have an activated gene, then we'll need to make a copy of that gene, or what's called a transcript. So, as we've shown before, messenger RNA, or mRNA, makes a complementary copy of that gene. Just like we did back here. We had our code of DNA, and then we entered the complementary bases that RNA would pair up with that sequence of DNA. So here we have DNA's code, and then we have the mRNA code. Then we divided that into triplets, and each of those, again, were called codons. Since RNA, the mRNA, is just a transcript of the one gene, and it's single-stranded. It's skinny enough to migrate from the nucleus to the cytoplasm or to a ribosome that's on an ER. The ribosomes are cytoplasmic granules that are composed of ribosomal RNA. Remember, that's more of the clumpy RNA, and enzymes. Transfer RNA, again, that's the clover leaf-shaped RNA. That's found in the cytoplasm, bound to an amino acid, and that's going to deliver those amino acids to the ribosome. So therefore we can say ribosomes assemble amino acids in their correct order, which is directed by the codons of mRNA, which was previously copied from DNA's code. That's how protein synthesis begins. We go from DNA to messenger RNA and eventually proteins. Transcription is the act of mRNA copying or transcribing DNA. This happens in the nucleus. Translation is the next step, which I've shown you before, where we actually start stringing together those amino acids in the correct order. We go from mRNA to protein. Most of this occurs in the cytoplasm at the ribosome. 10 to 15% of proteins are synthesized, though, in the nucleus. DNA is too large to leave the nucleus. That's why we need mRNA, because it can. Transcription, again, is carrying out the copying of the genetic code, one gene at a time, from DNA to RNA, and then carrying that out to a ribosome. We need some enzymes, though, to help us out. Remember, enzymes are usually proteins that do a specific function. RNA polymerase is one of those specific enzymes. This enzyme binds to DNA and then assembles the mRNA, basically binding to DNA and grabbing onto the mRNA nucleotides and stringing them together in the correct order. A base sequence like TA, 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 that's called a TATA box, informs RNA polymerase where to grab on to DNA. That's the beginning of the gene there. Grab on there, and that's where it's going to start. Then DNA unzips, just temporarily though. Remember, those hydrogen bonds are pretty weak, so it's able to do that. And so just about 17 base pairs at a time unzip. The code of DNA is then read, one base at a time, and it makes the corresponding mRNA. When it finds a C on DNA, it adds a G to the mRNA code or mRNA strand that it's making. And when it finds an A on DNA, it adds a U to the mRNA. Remember, RNA does not have any T's. RNA polymerase then can rewind the DNA helix back up behind it, and we can say our gene has been transcribed by several polymerase molecules at once. The terminator is the base sequence at the end of the gene that signals polymerase to stop. The pre-mRNA is then made. So we now have a strand of immature mRNA produced by transcription, but we're going to have to do some modifications to it before we send it out to the ribosome. First of all, the exons are the useful part that actually code for amino acids. So that's the sense portions of the immature mRNA. We're going to keep those. Those will go on to be codons that are translated into proteins. But remember, there's a lot of nonsense or junk portions to DNA that mRNA just copied. So we need to get rid of those parts. We need to remove those. Those are called introns. So in alternative splicing, the introns are removed by enzymes and spliced out, and then the exons spliced together, and then we have our functional RNA molecule. So one gene can code for more than one protein by doing different types or alternative splicing mechanisms. Here's some pictures of that. We have our DNA, then we've made our pre-mRNA, but it consists of exons and introns. So in one case here, our mRNA1, we keep the A, B, or the A, C, and the D, but in another circumstance, we keep the B, D, E. Another circumstance, we keep the A, E, F. So each of these can make different proteins. Each of these will have different codons that code for a different sequence of amino acids that can make 3 different proteins. All of this came from one area of DNA. Translation, then, is the process that converts that language of nucleotides into the language of amino acids. This happens at the ribosomes. Many of these occur in the cytosol. Some are on the surface of the rough ER, and some are just on the surface of the nuclear envelope. Again, ribosomal RNA looks very clumpy. It's kind of like 2 fists clumped together. There's a large part and a small part. The way they fit together helps to lock onto the mRNA strand, to read its codons, to be able to put together the right sequence of amino acids. So the mRNA begins with a leader sequence. This acts as a binding site for the ribosomal subunit. The large subunit attaches to the small subunit, and then the ribosome pulls the mRNA molecule through it, kind of like a ribbon, and it reads the bases as it goes. First, it reads the start codon. Remember, all proteins begin with that start codon of AUG. When we reach that, protein synthesis begins, and then we tack on that first amino acid called methionine. tRNA, remember, is the cloverleaf-shaped type of RNA that's in the cytoplasm, and it's attached to an amino acid, and its job is to bring that amino acid to the ribosome. So while the mRNA has the codon, we say that the tRNA has the anticodon. Here it is here. So this anticodon, this code right here, is going to match up with the correct codon and bring the correct amino acid to the ribosome. Then you can see how this all occurs in the right order based on that code that was initially copied from DNA. Each of these tRNA molecules are going to pick up a specific amino acid from that pool of free amino acids swimming around in the cytosol. That is going to take some ATP to do that. This is going to provide free energy for the peptide bond to be formed. Then the ribosome binds and holds the tRNA with its specific amino acid. The large ribosomal subunit then contains an enzyme that forms that peptide bond and links those amino acids together. Then the tRNA is released. It's going to go back out into the cytosol and find, again, its specific amino acid and do this all over again. Then the second, the tRNA will temporarily anchor the growing peptide chain. The ribosome will shift, and the third tRNA will bring its amino acid to the site. This is what it looks like under an electron microscope. We have our mRNA chain, and actually a bunch of ribosome units have grabbed on to that mRNA chain and are reading it. That way we're able to string together more than one amino acid chain at once from this one mRNA strand. This is called the polyribosome. One ribosome can assemble a protein of 400 amino acids in just 20 seconds. It's pretty fast. And 300,000 identical mRNA molecules can be undergoing simultaneous translation. So a cell can produce 150,000 protein molecules per second. So let's review that. Here's our double helix, DNA. Here's our gene. We're going to transcribe that. Then, after transcription, mRNA copies that. I can tell that this is RNA because you can see the U's, we can see the start codon, AUG, we can see a stop codon at the end. And if you like, you can pause the video and see if you can look up each of these codons using your amino acid chart that I gave you. We have our cloverleaf tRNAs who have picked up the proper amino acid, starting with the methionine. They look cloverleaf shaped. Remember, at one area of the cloverleaf we have the anticodon who's going to match up at the ribosome with the start codon. UAC matches with AUG. It's carrying its amino acid methionine. The ribosome will create that polypeptide bond to bond the next amino acid to it, alanine. There's our codon CGC on the tRNA will match up with GCG. And then the next one, and the next one until it reaches the stop codon, who doesn't code for anything but tells the ribosome to let it go. And then down below, we have our chain of amino acids. In reality, most polypeptides are hundreds of these long instead of just 6. Protein synthesis here gives us the primary structure of a protein. The right order of amino acids is that primary structure of a protein. But remember, we're not done yet. It must be folded into its secondary and tertiary structure. Remember, secondary structure is small local folding, like an alpha helix or a beta sheet. And tertiary structure is the whole molecule folding. And if we want to get to quaternary structure, then we've got to join different polypeptides together. Chaperone proteins are older proteins that pick up new proteins and guide the new ones in the folding into the proper shapes. Here's another look at how this happens, going from the nucleus where we have our DNA, out here to the ER where we have our ribosomes. Or remember, those ribosomes might also be floating in the cytosol. And then finishing up our protein and folding it in the Golgi complex. Then we might make a vesicle around that protein and send it out using a secretory vesicle. If you take this slide and on your PowerPoint and play it with each click, it'll take you right through the process as well, click by click, from DNA to mRNA, out to the ribosomes, stringing together our polypeptide with amino acids, folding it here, finishing it in the Golgi apparatus, making a vesicle, and secreting it into the bloodstream, and then it's on its way. Proteins to be used in the cytosol are likely to be made on the free ribosomes in the cytosol, but those that are destined for packaging and to be sent somewhere else are usually then assembled on the rough ER and then sent to the Golgi for packaging. The entire polyribosome migrated to the rough ER and then docks on its surface. And then the assembled amino acid chain is completed on the rough ER and then sent to the Golgi for final modification. The vesicles then can fuse and unload the proteins into the Golgi cisternae, and then the Golgi complex can further modify the protein and fold it. The process of using an excretory vesicle to get rid of that protein and send it on its way is called exocytosis. Genes can be turned on and off from day to day, from hour to hour, because their products may or may not be needed. It's a waste of energy to make products if we actually don't need them at the time. Many genes can be permanently turned off in any given cell. Like, for instance, if we have a gene for hemoglobin, we wouldn't want to be making that protein in, like, say, a cell of your eye. That would be useless. Or a digestive enzyme. We don't want to make those proteins where they don't belong. We only want to make digestive enzymes in cells that are in our stomach or in our small intestine. There's several ways to turn genes on and off. Let's consider a woman who's just given birth to her first baby. In the next few days, the hormone prolactin stimulates cells of our mammary glands to synthesize the various components of breast milk. This includes the protein casein. That's something her body has never synthesized before. How is the gene for casein turned on at this point in her life? Well, prolactin will bind to its receptor, which is a pair of proteins in the plasma membrane of a mammary cell. Next, the receptor triggers the activation of a regulatory protein that's called the transcription activator. Next, the regulatory protein moves into the nucleus and binds to the DNA near the casein gene. This binding enables RNA polymerase to bind to the gene and then transcribe it, producing the mRNA for casein. Next, the casein mRNA moves to the cytoplasm and is then translated by ribosomes on the rough ER. Then the Golgi complex packages the casein into secretory vesicles, and those secretory vesicles release the casein by exocytosis, and it becomes part of the milk. You can follow that process with this diagram. All of the steps are numbered as they are in the slides above. Cells, of course, make more than just proteins. They also synthesize carbohydrates like glycogen. They also make fat, steroids, phospholipids, pigments, and many other compounds. And there's no genes for these. But then how would their synthesis be possible? Well, their synthesis is said to be under indirect genetic control. How? They're produced by enzymatic reactions, and enzymes are proteins encoded by genes. So, for example, testosterone production. Testosterone is a steroid, which is a lipid. So a cell of the testes will take in cholesterol and then use enzymes to convert it to testosterone. And this will only occur when genes for that enzyme are activated. So genes can greatly affect such complex outcomes as behavior, aggression, and even sex drive. So let's see if you can answer these questions. Now that we've completed this section, you can pause the video and see if you can answer them before I do. Number 1, define a gene, genetic code, codon, and an anticodon. Answer. A gene is an information-containing segment of DNA that codes for the production of a molecule of RNA, which in most cases will go on to play a role in the synthesis of one or more proteins. The genetic code is a system that enables the 4 nucleotides of DNA to code for the amino acid sequences of all your proteins. A codon is every 3 letters of mRNA, which was transcribed from DNA, and a codon will code for a specific amino acid if it's a useful one. And an anticodon is the opposite of that codon, the complementary code that will match up with the codon that's carried on the tRNA. Number 2, describe the roles of RNA polymerase, ribosomes, and tRNA in producing a protein. Answer: RNA polymerase is an enzyme. It's located in the nucleus of the cells. It binds to DNA at a certain site, usually TATA box, TATA box, and then it allows the RNA nucleotides to be assembled complementary to the code in DNA. A ribosome is kind of a double clump of ribosomal RNA plus enzymes that's going to be responsible for grabbing onto the mRNA to assemble proteins. They can be found on the ER or free floating in the cytoplasm or on the nuclear envelope. tRNA is the cloverleaf shaped RNA floating around in the cytosol, which grabs onto specific amino acids according to its anticodon that it contains. And then it'll bring those amino acids to the ribosome. Number 3, what's the difference between genetic transcription and translation? Genetic transcription is when RNA polymerase creates the mRNA, which is complementary to DNA's code to that gene. And translation is the process of going from mRNA to a chain of amino acids, which will make a protein. Number 4, summarize the processing of a protein. I'm going to go back to this picture for that one. In the processing of a protein, we have the primary structure where we string the correct order of amino acids together at the ribosome, and then we send it on further to do the local folding, which is the 2nd degree structure, and then we do whole molecule folding, which is the 3rd degree structure, and join it with other polypeptide chains where we have finished our protein, and then possibly put it into a vesicle and secrete it to where it needs to go. Describe a way that a gene can be turned on or off at different times in a person's life. In our notes, we looked at the way prolactin does this. So in this sense, the hormone triggered intracellular reactions that activated a regulatory protein. Then that protein binds to DNA near the proper gene that we want to transcribe, and it enables the RNA polymerase to produce an mRNA for that gene. Then that mRNA is translated to synthesize the protein, and then we have made a protein that, like in the case of a mother making milk for the first time, she's making a protein she's never made before in her life. Number 6, considering that genes can only code for RNA or proteins, how can the synthesis of non-protein substances like carbs or steroids be under genetic control? Well, if we remember that proteins and enzymes are the workhorses of the body that get certain jobs done and make reactions happen, like making carbs and steroids, remember that proteins and enzymes are coded for by DNA. So you have to have the right proteins and enzymes available to make these other substances, and that's how they're indirectly controlled. Here's the learning outcomes for our next section. You can pause the video and read those over. We're going to take a look at DNA replication and the cell cycle. Before a cell divides, it must duplicate its DNA so that it can give a complete copy of all those genes to the daughter cell, because that daughter cell, the individual cell, will need to be able to make the right proteins and enzymes, RNA, so it must have a copy, a complete copy of that DNA. Since DNA controls all the cellular function, that replication process needs to be very exact. Luckily, we have the law of complementary base pairing. So if DNA has an A on one side, we know the other side of DNA will be a T, and then C's with G's. This actually enables the cell to reproduce one strand based on the information of the other. So first, the double helix of DNA will unwind from its histone proteins. This is a permanent unwinding, unlike with DNA transcription, that was just temporary. DNA replication is a permanent unwinding. An enzyme called DNA helicase will open one short segment of the helix at a time to expose those nitrogenous bases. That just involves breaking those weak hydrogen bonds holding the helix together. This creates a replication fork. This is the point where the DNA is opened up like two separated halves of a zipper. Then DNA polymerase molecules will move along each strand, and it will read those exposed bases and match up complementary free nucleotides. The two separated strands of DNA are copied by separate polymerase molecules proceeding in opposite directions. The polymerase molecule will move toward the replication fork and makes a long, continuous new strand of DNA. The polymerase molecule moving away from the replication fork will make short little segments of DNA at a time, but that's okay because another enzyme called DNA ligase will join them together later. So from the old parental DNA molecule, we'll get two new daughter DNA molecules. This is called semi-conservative replication. Each daughter DNA strand will consist of one newly synthesized helix and one old. Here's one picture of that happening. The big green things are the DNA polymerase. The orange thing is the DNA helicase. And DNA ligase on, this is called the lagging strand here, putting together the little pieces of DNA, making sure they're sealed up into one long continuous strand. New histones are synthesized in the cytoplasm. Millions of histones are transported into the nucleus within a few minutes after DNA replication. Then each new DNA helix wraps around them to make a new nucleosome, and each DNA polymerase works at a rate of 100 base pairs per second. This would take weeks for one polymerase to replicate one chromosome. So thousands of polymerase molecules are at work simultaneously at any given time on each DNA molecule. So all 46 chromosomes can be replicated in 6 to 8 hours. Unfortunately, DNA polymerase can make mistakes. There's multiple modes for correction of these replication errors, though. It double checks the new base pair and tends to replace the incorrect, biochemically unstable pairs with a more stable correct pair, should that happen. And this results in only one error in every 1 billion bases that are replicated. Unfortunately, some of them are not caught. That's called a mutation. A mutation is a change in the DNA structure or the order of the A, T, C's, and G's due to replication errors or other environmental factors. Radiation, UV light, viruses, chemicals, pesticides, things like that can actually cause mutations as well. Some mutations can cause no ill effect, while others can kill the cell. Some can turn it cancerous, or some can cause genetic defects in the future generation. Here's a picture of DNA being copied in this semi-conservative process. DNA visualized here is unzipped. The blue is the old strand, the parent strand, while the orange is the new daughter strand. So when we're done, we'll have two molecules of DNA. Each will have an old blue parent strand, and each will have a new daughter strand, which is the orange part. This is called semi-conservative replication. Here's a picture of the helicase enzyme unzipping DNA, making a replication bubble right here and moving in both directions. Here's a picture of replication gone further. We have our replication bubble, and our DNA polymerase is going to start here, and it's going to move in both directions. The one that goes in this direction goes continuously, making a leading strand. The one that goes in this direction has to do it in segments because of some of the rules that it has to follow. That would be beyond this course to discuss. But it does it in segments, but eventually these segments in the lagging strand will have to be sealed together with DNA ligase. Then we have this side also being replicated at the same time, with DNA polymerase going this way, making the leading strand continuous, with the other side making it in segments. Here's a picture of what it looks like under an electron microscope. If we consider this as one long strand of DNA, each of these areas that you can see here is a replication bubble. And you can see this particular DNA strand has 4 replication bubbles going on at one time. You can match them up with the picture off to the right. Here's the first one, the second one, the third one, and the fourth. In this diagram, we can see some proofreading that DNA does, that the polymerase does, I should say. We have an unstable pairing, so that is sliced out and then replaced with a more stable pairing, and then sealed with DNA ligase. Next, let's take a look at the cell cycle. Replication of DNA is going to have to take place somewhere along the lines here of a cell cycle. The cell cycle is the cell's life cycle that extends from one division to the next. The G1 phase, or what's called the gap 1 phase, is the interval between cell division and DNA replication. We can see it right here. This is when enough materials are accumulated which are needed to replicate the DNA. It's also a phase in a cell's life cycle where it's doing normal cell things like making energy and making proteins and such, but it's also growing and getting enough materials collected for when it enters the next phase, which is S phase, when replication occurs. S is for synthesis. The G2 phase is called the gap 2 phase. That's right here. The G2 phase is a lot like the G1 phase, but it happens after synthesis. This is between DNA replication and cell division. Centrioles are finished with their duplication. They're going to help to control the cell division when the time comes. Enzymes are synthesized that control cell division and any repair that needs to be done because of DNA replication errors, that's done as well. The M phase is right here. That's the phase of mitosis. Actually, I like this diagram better for looking at the M phase right here. It shows you how short the M phase is compared to all the others, the G1, the S, and the G2. The M phase is when the cell replicates its nucleus. M phase also stands for, or is similar to the word mitosis, which you may have heard before. Interphase is all of this time, which includes the G1, the S, and the G2 phases. So we have interphase, and then we have the M phase, when the cell is actually dividing. Interphase is just like normal cell activity plus the synthesis. M phase is when a cell is actually dividing. The cell cycle and how long it takes will vary between cell types. Some cells go through it often, like for instance your skin cells, your stomach cells, but some cells really don't after you're born or after growth, such as nerve cells. Mitosis is the cell division in all body cells except egg and sperm. They undergo something just a little bit different. The functions of mitosis is the development of the individual from one fertilized egg to about 40 trillion cells. So you have to go in 9 months from one fertilized egg to about 40 trillion cells. In that time, you have to grow all your tissues and your organs. You have to replace cells that die and repair damaged ones. Within mitosis, there's 4 phases. They are prophase, metaphase, anaphase, and telophase. Here's a diagram showing phases and a little summary of what happens during each one, and we'll go through that. If we were to look at a cell in prophase under a microscope, here's what we would see. We would see the chromosomes starting to shorten and thicken and coil into compact rods. This would be making it easier for us to actually see the chromosomes in the cell. Doing this makes it easier to distribute DNA to the daughter cells than it would be with just the chromatin alone. This happens to our 46 chromosomes, 23 from mom, 23 from dad. We'll know that they've been duplicated because the chromosomes will appear as the doubled sister chromatid form. The nuclear envelope will start to disintegrate and will release those chromosomes into the cytosol. We'll see the centrioles migrating to opposite sides and microtubules sprouting, creating what's called the mitotic spindle or spindle fibers. Will look a lot like this. We can see the mitotic spindle. Here's the centrioles right there. Nuclear envelope is starting to go away, and we have our chromosomes appearing as sister chromatids visible within the area. Here we see 4 chromosomes instead of the 46 that a normal human cell would have. Spindle fibers will begin then to tug the chromosomes back and forth until they line up in the middle of the cell. And when that happens, we know we have reached metaphase, because all those chromosomes will be lined up at the cell equator. They'll oscillate slightly and await the signal that stimulates each of them to split. The mitotic spindle is a lemon-shaped array of spindle fibers. Kind of looks like a spider web. The long spindle fibers, or microtubules, will attach to the chromosomes, and the shorter microtubules, called the aster, will anchor the centrioles to the plasma membrane at each edge of the cell, or each end of the cell. The spindle fibers will keep pulling. They'll attach to those kinetochores of the chromosomes, keep pulling until finally they pull the sister chromatids apart from each other. When we see them starting to pull apart, we'll know we have reached anaphase. During telophase, the chromatids will cluster on each side of the cell. Rough ER will produce a new nuclear envelope around each cluster. The chromatids will begin to uncoil and form chromatin again. The mitotic spindle will break up and vanish, and each nucleus will form nuclei again, indicating that it's already begun making RNA and preparing for protein synthesis. Let's go back to this picture and look at this again. We have prophase, we have metaphase. That's maybe one of the most easily distinguishable phases of mitosis, where we see all of the chromosomes lined up at the equator. Then we see them pulled apart during anaphase, kind of like they're being lassoed and pulled forward. Telophase, we actually see the nucleus has now divided. Even cytokinesis has begun, where the cytoplasm is dividing as well, and we'll end up with two daughter cells who will have complete copies of the genetic code. Again, cytokinesis is the division of the cytoplasm. This is achieved by our motor proteins, myosin and actin. This creates a cleavage furrow, and the cell will eventually pinch in two. Cells will only divide when they've got enough cytoplasm for two daughter cells, when they've replicated their DNA, when they have adequate supply of nutrients, and they're stimulated by a growth factor, a chemical signal. The neighboring cells might die or open up or leave space in a tissue, and that might also signal for a cell to divide as well. Cells stop dividing when they are snugly in contact with neighboring cells, when they have enough nutrients, or when they don't have enough nutrients, or when growth factors are withdrawn, or when they undergo contact inhibition, the cessation of cell division in response to contact with other cells. Cancer cells will divide despite all of this. All right, let's see if you can answer these questions at the end of this segment. You can pause the video to see if you can answer them first before I do. Number 1, describe the genetic roles of DNA helicase and DNA polymerase. And what's the function of RNA polymerase? DNA helicase unwinds the double helix of DNA, while DNA polymerase adds in the appropriate DNA nucleotides to replicate DNA. A's with T's, C's with G's. So then the function of RNA polymerase is not during replication. This is during transcription, when a mRNA transcript is made during protein synthesis. Number 2, explain why DNA replication is semi-conservative. Well, remember when DNA is unzipped, then bases are filled in. Each new strand will contain an old parent, strand of DNA and a new daughter strand of DNA. Number 3, what's a mutation? A mutation is when a wrong nucleotide gets put in during replication or even during transcription. So why are some harmless and others lethal? A mutation could occur in a non-coding section of DNA, in a part that doesn't code for any amino acid. In that case, it wouldn't matter. If it does occur in one of the codons, then you'll get the wrong amino acid put in place, and then it'll make that protein or enzyme non-functional. Number 4, list the stages of the cell cycle and summarize what occurs in each. I'm going to go back to this picture to help with that one. Prophase, we start to see chromosomes because the DNA wraps around those histone proteins and condenses. The nucleus goes away and the mitotic spindle begins to be set up in prophase. In metaphase, our mitotic spindle is complete with the centrioles on opposite sides, the mitotic spindle set up, and our chromosomes are at the equator. In anaphase, those chromosomes are pulled apart. Telophase, the nuclear envelope begins to form again, and because the nucleus has now divided, and cytokinesis begins to happen as well. And our chromosomes will start to decondense back to chromatin again. Five, list the stages of mitosis. Oh, we already did that. Oh, did I answer these wrong? I might have answered them wrong. I just answered number 5, listing the stages of mitosis and what happens during each. Number 4, I'm going to go back to that one. What are the stages of the cell cycle and what happens during each? For that one, I'm going to have to go back to this diagram. The stages of the cell cycle, G1, that's when a cell is growing and getting enough materials for the S phase, synthesis, when DNA replicates, and the G2 phase, getting ready for mitosis. Alright, here's the last section of our chapter. You can read the expected learning outcomes here. Let's take a look at chromosomes and heredity. Heredity is the transmission of genetic characteristics from parent to offspring. A karyotype is a chart of 46 chromosomes laid out in order by size and other physical features. I'm going to go to this picture. This is called a chromosome spread. This is from a cell who's already undergone the S phase of the cell cycle. So each of these chromosomes occur as sister chromatids. They're duplicated. They've also been stained with gemcistane, and the stain makes the chromosome pick up this banding pattern. So someone with a trained eye then can match the chromosome pairs. Remember, for each chromosome, we get one from mom, one from dad that occur in pairs. They'll match them up by the banding pattern and the size to create this. And this is called a karyotype. We can see how chromosome pair number 1 is the largest. They have the same banding pattern, about the same size. Chromosome pair 2, 3, all the way down to our smaller ones here, 20, 21, and 22. All of these chromosomes are called autosomes. The last pair are called the sex chromosomes. In this case, for the last pair, we can see that mom gave an X and dad gave an X chromosome, and this will create a female, a baby girl. Here's another spread of chromosomes. We put them together, we get this combination. In this case, This will create a baby boy because mom gave an X chromosome and dad gave a Y chromosome. With all these other chromosome pairs, these are called homologous pairs. They have the same genes on them, maybe different alleles though. Remember, an allele is a different version of a gene, such as blood type. You might get the A allele or the B allele or the O allele. As for the X and Y chromosome, they are not homologous. They have different genes on them. You can see the Y chromosome is a very small chromosome. There's only about 28 or so genes on that chromosome. Most of them have to do with male gender. A human karyotype is said to be diploid, that we have 2 copies of each chromosome in all of our what are called somatic cells. Somatic cells are regular body cells that have 46 chromosomes or 23 pairs. A haploid cell, though, that's our sperm and our egg. Haploid cells are also called germ cells or gametes. They have only half the chromosomes. Fertilization then will restore the diploid number again. So that's why we say you get half of your chromosomes from mom and half from dad. Half of them were in the egg cell and half were in the sperm. A locus is the location of a particular gene on a chromosome, and an allele is a different version of a gene at the same locus on 2 homologous chromosomes. Genes can come in dominant and recessive alleles. The dominant one, if we're doing genetics problems, we usually represent that one by a capital letter. A dominant allele will always cover a recessive allele. It produces a protein responsible for a visible trait. The recessive allele, if we're doing genetics problems, usually gets the lowercase letter. Recessive alleles are only expressed if the dominant allele is not present. So for instance, cleft chin, we've got a Punnett square here. I believe this is actually John Travolta's chin. If I remember right, he's famous for having a cleft chin. It looks like cleft chin is dominant. So you can either receive 2 alleles for the dominant gene and have a cleft chin, or you can be what's called heterozygous and have a dominant and a recessive. Again, the dominant always overrides the recessive. So we have the cleft chin, even though this person is carrying the recessive allele. The only way to get the non-cleft chin is to have 2 recessive alleles for that. So if you get this combination, you're called homozygous recessive. If you get this combination, big C, you're homozygous dominant. And if you get one of each, you're heterozygous. That combination that you get is called your genotype. So in this case, we have 3 different genotypes: big C, big C little c, or little c, little c. The phenotype is actually the express trait. So for instance, to let you get an idea of that, if you look at the person with the cleft chin, it's hard to tell what the genotype is by looking at the phenotype. The phenotype is the cleft chin. But how do you know the genotype? Because with having a cleft chin, your genotype could be big C, or it could be big C, little c. However, with having a phenotype of a non-cleft chin, you know that in order to have that person must be little c, little c. Genetic counselors can perform genetic testing and can refer clients for tests and advise couples on the probability of transmitting certain genetic diseases, and then can help them cope with that disease. A Punnett square, like this one, can help us make those determinations on what is the probability that you'll get that trait. Cystic fibrosis is an example of a homozygous recessive disorder. So you would need two little letters, two recessive alleles, in order to get cystic fibrosis. A genetic counselor could help parents figure out the probability of giving their child cystic fibrosis if both of them are heterozygous carriers. And then if they do give the child cystic fibrosis, they can help the parent deal with that. The gene pool is the collective genetic makeup of a population as a whole. Multiple alleles is when there's more than two allelic forms for a trait. For instance, there's multiple alleles for the ABO blood group. You could get the A allele, the B allele, or the O allele. In this case, because there's multiple alleles, they use a big I here, or a little I, as a representative of the gene for blood type. And then the A, B are both dominant. O is recessive. When two genes are dominant together, meaning if you get both of them and they're both expressed together, that's called codominance. So if mom gave you an A allele and dad gave you a B allele, you would have type AB blood. However, if, let's say, mom gave you an A and dad gave you an O, because A is dominant over O, you would have type A blood, and you would be a carrier of the O allele, but it would not be expressed. Incomplete dominance is when you get two alleles and there's a blending of the traits, such as in certain flowers, there's a red pigment and then white. If you got both of those alleles, you would end up with pink. So that's more of a blending. As for the codominance with the ABO blood group, your red blood cells would have both the A and the B glycoproteins on the cell surface. O is when there's neither of those glycoproteins. Polygenic or multiple gene inheritance is a phenomenon in which genes at two or more loci, or even on different chromosomes, contribute to a single phenotypic trait. Human eye and skin colors are good examples of polygenic traits. Pleiotropy is a phenomenon in which one gene produces multiple phenotypic effects. For example, about 1 in 200,000 people have a genetic disorder called alkaptonuria. This is a mutation on chromosome number 3 that blocks the breakdown of the amino acid tyrosine. This results in the accumulation of an intermediate breakdown product that's in the body, and it harms connective tissue and causes degeneration of cartilages. Among the multiple phenotypic effects of that disorder are darkening of the skin, darkening and degeneration of cartilage in places like the ears and knees and the intervertebral discs, and darkening of urine when it stands long enough to oxidize, discoloration of teeth, and even the whites of the eyes. It even causes arthritis in the shoulders, hips, and knees, and can damage the heart valves and prostate gland. Another well-known example of pleiotropy is sickle cell disease, which will be detailed in a later chapter. Some genes are carried on the X or the Y chromosome. Those are called sex-linked traits. These are traits carried on the X and Y chromosome. Therefore, it kind of changes the probability in getting certain disorders. Recessive color blindness is an allele that's on the X chromosome, but remember the X and the Y are not homologous, so this gene is not on the Y chromosome. So let's say that a little c means that you have the allele for color blindness. If you're a female and you have at least one big C, which is dominant and normal, it'll cover up the little c. So to be a female, to have this colorblind disorder, you have to get two little c's. One must come from your mother and one must come from your father. In this case, we have one X chromosome here from one parent with a big C. The other parent gave the little c. They're just a carrier. They do not have color blindness. But a male who gets the X chromosome from their mother that has the little c on it, they will be colorblind. So you can see where you would have in a population more males who would have color blindness than you would females. In order for females to be colorblind, they must get it from both parents, but males only need to get it from one parent. Again, that's called sex linkage. Penetrance is the percentage of a population with a given phenotype that actually exhibits the predictive phenotype. If 80% of people with the polydactyly allele actually exhibit extra digits, the allele has 80% penetrance. Polydactyly is the presence of extra fingers and toes. It's actually a dominant gene. The role of the environment. No gene can produce a phenotypic effect without nutritional and other environmental input. So to be human, we need both the genetic recipe and we need the other ingredients from our environment as well. For instance, brown eye color requires an amino acid called phenylalanine from our diet to produce melanin pigment. A common misconception is that dominant alleles must be more common in a gene pool than recessive. But that's not always true. Some recessive alleles, like the blood type O, are most common. Some dominant alleles, like polydactyly and the blood type AB, which is actually codominant, those are rare in a population. A newer growing field is called epigenetics. Epigenetics means heritable changes that are beyond the level of the genome. We're just beginning to study and understand this, but it's not always just about the code of DNA or mutations. The way DNA winds around chromosomes and how functional groups like methyl groups are CH3 groups, how those can get attached to DNA and make them wind tighter or less tight and allow enzymes to get in there and transcribe the codes. That also controls our genes as well. We must now view heredity as entailing more than just the genes that are passed from one generation to the next. A related field to genetics is the study of cancer or oncology. It deals with both benign and malignant tumors, but only malignancies are called cancer. The word literally means crab. Hippocrates was the first to use that word in that way upon seeing a breast tumor with a tangle of blood vessels that reminded him of a crab's outstretched legs. Energy-hungry tumors often stimulate such that ingrowth of blood vessels. That's called tumor angiogenesis. Cancers are often caused when cell division goes wrong. when cells defy the cell cycle and they divide anyway uncontrollably. Cancers are named for their tissue of origin, such as a carcinoma is in the epithelial tissue, lymphomas are in the lymph nodes, melanomas are in pigment cells of the epidermis or the melanocytes, leukemias are in the blood-forming tissues, and sarcomas are in the bone or connective tissue or the muscle. A carcinogen is any environmental cancer-causing agent. Carcinogens can change or mutate our genes, which then makes those cells not follow the cell cycle, and it makes them grow uncontrollably into these big masses that can start to invade other tissues. Radiation, chemicals, and viruses can actually act as carcinogens that mutate our own DNA. Only about 5 to 10% of cancers are hereditary. Oncogenes cause cell division to accelerate out of control. Basically, they're going against the cell cycle. In the cell cycle, chemicals would normally control the growth of a cell, letting it know when it should divide and when it shouldn't. But if those genes that control that get mutated, like these oncogenes, then it'll divide out of control, despite what the cell cycle says. Tumor suppressor genes normally are there to stop this from happening, or to make a cell commit suicide if it starts to grow out of control. But if these genes get mutated as well, then there's nothing to stop it. Cancer seldom results from just one mutation. It usually requires 5 to 10 at different gene loci. It takes time for many of these mutations to accumulate, so that's why older people tend to have cancer occur more than the young. Cancer claims the lives of about 1 to 5 Americans and is almost always fatal if not treated. Malignant tumors replace functional tissue in the vital organs, and they steal nutrients from the rest of the body, sometimes causing a severe wasting away called cachexia. They weaken one's immunity, and they open the door to other opportunistic infections that a healthier person could usually ward off. They often invade blood vessels, lung tissue, and brain tissue, with consequences like hemorrhage and pulmonary collapse, seizures, and coma. Mortality usually results not from the original tumor itself, but from the metastasis, the spreading of it. Cancer is usually treated with surgery, chemotherapy, radiation. A lively area of cancer research today is the development of drugs to starve the tumor by blocking the tumor angiogenesis. Let's see if you can answer these questions in our last section. Why must the carrier of a genetic disease be heterozygous? Well, the carrier carries the recessive allele for that specific trait, such as big C, little c. The little c is the recessive allele. The person doesn't actually have the disease because the normal dominant gene overrides the bad recessive one. Number 2, why can't a person's genotype always be determined from their phenotype? Well, that goes back to question 1. If somebody is heterozygous, they might be carrying the recessive allele, but you won't know that. Number 3, a blind man can inherit color blindness from his mother, but not his father. Explain that. Well, color blindness is a trait, a gene that's carried on the X chromosome. And if you're a man, you inherit one X chromosome, and that comes from your mother. But from your father, you get a Y chromosome, which is not homologous to the X. and so therefore does not have any genes for color blindness on it.